Okay, so hello everyone. So in last night's video, we learned a little bit about why cells need to reproduce. And we saw if we had a single-celled organisms, cells reproduce to create more of themselves, to create more organisms. And if we had a multicellular organisms, a cell would reproduce to either grow, have the organism grow, or to repair damaged cells, any of those things. So today's video is going to focus on how cells divide. Yesterday you looked at the why cells divide, and today we're going to look at how cells divide. So the process by which cells divide is called cell reproduction, or the cell cycle. And the cell cycle is basically the series of activities that a cell undergoes when it's dividing or when it's preparing to divide. So if we look at this little picture over here, the cell cycle is divided into two pieces. So this first piece right here is interphase, and this is basically when the cell is getting ready to divide. And then the second phase is called the mitotic phase, which is this little yellow portion right here. And this is when the cell is actually dividing. So we have interphase, when the cell is getting ready to divide, and then mitotic phase, when the cell actually is dividing. So let's break these two phases down a little. So if we look at this picture, we see that this yellow portion, which is supposed to represent interphase, is actually divided into three sections. There's G1. This is where the cell is going to get bigger. Because if we're going to take one cell and turn it into two, the first thing that has to happen is the cell has to get bigger. Otherwise, with each division, we're just going to get smaller and smaller cells. The next phase is S phase, or synthesis, and this is basically where we copy the DNA. Remember yesterday, in yesterday's video, we talked about how DNA has to be copied before the cell can divide in order to make sure when we have two new cells, we have a copy of the DNA for each cell. And then the other part of interphase is this G2, where the cell grows a little bit more. So that's interphase. That's when the cell's getting ready to divide. And then the mitotic phase, this little blue portion right here, is further divided into 5 more categories that we'll talk about a little later in the video. So let's look at interphase first. So interphase is the longest phase of the cell cycle. In fact, it makes up 90% of the cell cycle. So that means basically 90% of your cell's lives are just getting ready to divide. Like we saw in the last slide, interphase has 3 phases, G1, S, and G2. So G1, that stands for growth 1. We're going to get the cell to grow. We're going to make enough enzymes so that way we have, 2 copies of each enzyme. We have enough for each of my 2 new cells that I'm going to be forming. We copy all of our organelles, because again, we're going to be forming 2 cells. So we need to have enough organelles for both. Then S phase, S stands for synthesis. Remember, we've seen this word many times this year so far. So synthesis, that means to build something. So in the synthesis phase, we're going to be building a new copy of our DNA. We're going to be going from chromosomes that look like this, so just one copy of the chromosome. We're going to replicate them. So then our chromosomes are going to take this X shape, like you saw in the video from yesterday. Then once we have 2 copies of each chromosome, now we're ready to make my 2 cells. So then finally we get to the last phase of interphase, which is G2, where the cell gets just a little bit bigger. Okay, so that's interphase. So the other 10% of the cell cycle is made up of the mitotic phase. And remember I said earlier, the mitotic phase is when the cell actually divides. So this mitotic phase is split into 2 distinct sections. I have mitosis. This is when my nucleus is going to divide. So and everything in the nucleus. So all of my chromosomes, I'm going to get half of my chromosomes to go to one new nucleus, and the other half of my replicated chromosomes to go to my other new nucleus. So I've created 2 nuclei that I will then use to make my 2 cells. And then finally, the mitotic phase has a second part called cytokinesis. And this is basically when the cytoplasm is split in half and everything in the cytoplasm is split in half, basically. Okay. So, of mitosis and cytokinesis, we're going to look at mitosis first. So, like I said on the previous slide, this is the process by which the nucleus is divided into 2 identical nuclei. And, like we saw earlier in the year in the cell unit, the most important thing inside the nucleus is the chromosomes. So for example, humans have 46 chromosomes. So during S phase, during interphase, I'm going to replicate my chromosomes. So I go from 46 chromosomes that look like this, they're just one copy, to 46 chromosomes that are replicated. And then during mitosis, I'm going to basically split each chromosome in half and send half to one nucleus and half to the other. So then I'll eventually have two cells that have 46 chromosomes. And we call the two new cells that I'm creating, we call those daughter cells. The new cells are called daughter cells. Oh, I think I hit the wrong button. Okay. So kind of to reiterate this one more time, so during interphase, and more specifically during S phase, I'm going to replicate my chromosomes. So I'm going to make an identical copy of it. So now, but then at the beginning of mitosis, I'm going to start with that replicated chromosome, but at the end, I'm going to end up with two cells, each with one chromosome in it. So mitosis itself, the splitting of the nucleus, can be broken down into 4 phases. We have prophase, metaphase, anaphase, and telophase. And then those 4 phases are then followed by the splitting of the cytoplasm, or cytokinesis. So this should say followed by cytokinesis. I don't know why this word is way over here. That should be over there. So we're going to look at these 4 phases first. These are where the nucleus itself splits, and then we'll look at cytokinesis, when the cytoplasm splits. Okay. So this picture is just to remind you that before we get mitosis, we have interphase, so G1, S, and G. And then the first phase of mitosis is going to be prophase. And the way I think of it is that prophase starts with a P and preparation starts with a P. So really prophase is when the cell is preparing to divide. This is kind of like the preparation phase. So the first thing that's going to happen is we're going to condense our chromosomes. So in yesterday's video, we talked about how DNA can exist in 2 forms. It can exist in its spaghetti form, the chromatin form. But you can kind of think that this would be hard to deal with. If it's in this form, it's going to be hard to separate without breaking it. So the first thing that happens is prophase is my spaghetti-like form of my DNA, my chromatin, condenses into nice, organized shapes called chromosomes. And just as a reminder, we saw this vocabulary yesterday, but each copy, each little V is called a chromatid, and they're held together in the middle by what's called a centromere. So over here, this picture over here, this would be one chromatid, so that's one copy of my DNA. This is a second chromatid, and my 2 chromatids are held together in the middle by this centromere. We saw that vocabulary yesterday. So the first thing that happens in prophase is my DNA condenses into chromosomes, and then the second thing that happens is the nuclear envelope and the nucleolus break down. If I'm going to divide my nucleus in half, I have to get rid of the nuclear envelope and the nucleolus, otherwise they'll just be in the way. The last thing that happens in prophase is that I build and assemble the machinery that's going to actually separate the chromosomes. So the machinery that separates the chromosomes are in a complex called centrosomes. So centrosomes are the complex that contains the machinery that's going to separate the chromosomes. One thing that the centrosomes contain are called centrioles. These are the organelles that build the spindle, build these little yellow fibers that are going to attach to the chromosomes and pull them apart. So these centrosomes, they appear and they start moving to the 2 sides of the cell so they can separate the chromosomes. And then the mitotic spindle also forms. The mitotic spindle are these yellow little fibers in this picture here that are going to attach to the chromosomes and then pull them apart. So here I have a picture of prophase. You can see my nuclear envelope, this purple thing here, is starting to break down. My centrioles and spindle are starting to form, and my chromosomes, my yellow things in that picture there, are starting to condense, get into their chromosome form. And then by the end of prophase, my spindle is totally formed. So you can see my centrioles have formed the spindle and they've attached to the chromosomes. The nuclear envelope is almost all gone, and my chromosomes are totally in their condensed form. And here is a picture of a cell actually going through prophase. So you can tell it's prophase because the chromosomes are in their condensed form. They're not all spaghetti-y. They're in these little dense X's you can see in the picture there. Okay, so that was prophase. The second phase of the mitotic phase is called metaphase. And I like to think that metaphase starts with an M and middle starts with an M. So metaphase is the phase where all my chromosomes are going to line up in a straight line right in the center of the cell, right in the middle of the cell. So as you can see in the picture, in this phase, my centrosomes are totally formed and my spindle is attached to my chromosomes. But the hallmark of metaphase is you can see all my chromosomes are lined up in a straight line right down the middle. And the reason cells do this, we want to make sure that 1/2 of each chromosome goes to each new cell. And if the chromosomes are all over the cell, it's going to be hard to accomplish this. But if they're all lined up down the middle, it's easier for the cell to kind of break the chromosomes in half and take 1/2 to one side and 1/2 to the other. So a new vocabulary word for you is this word kinetochore. Oops, hold on. There we go. So if I have a chromosome, oh, this is not working. Sorry, technical difficulties. There we go. So if I have a chromosome, so here's my chromosome. So my two sister chromatids connected by my centromere here. So in order to split this chromosome in half, I have to pull 1/2 one way and 1/2 the other way. And the way this works is my spindle fiber attaches to the chromosome, and then they virtually pull in opposite directions to pull it apart. And the spot right here where the spindle fiber attaches, that's called the kinetochore. It's the spot on the centromere where the spindle fiber is going to attach. There we go. Okay. So that's metaphase. You can see I have my chromosomes in a nice straight line down the middle, and my spindle fibers are attached to the centromere at the kinetochore. So this is an actual picture of a cell in metaphase, and again, you can tell it's metaphase because all the chromosomes are lined up right down the middle, and you can see the spindle are attached to the chromosomes. So the next phase is called anaphase, and I think anaphase starts with an A, and the word apart starts with an A. So during anaphase, I'm going to be pulling my two chromosomes apart. So basically what happens is the centromere, the part that holds the two halves of the chromosome together, that breaks down, and then the spindle fibers shorten, which effectively pulls 1/2 of the chromatids to one side and 1/2 to the other. So basically I'm creating two distinct groups of chromosomes. So in order for me to separate my in order for me to separate my chromosome, I have to break the part that holds the two halves together, and then I'm going to pull 1/2 to one side of the cell and 1/2 to the other. That happens during anaphase. So here's a picture of an actual cell going through anaphase, and you can tell it's anaphase because I have 1/2 of my chromosomes going to one side of the cell and 1/2 being pulled to the other side. And again, you can see that here. So then finally, the last phase is telophase, and I think that telophase begins with a T, and 2, the number 2, begins with a T. So in telophase, I'm going to form two distinct nuclei. And you can kind of think of telophase as the reverse of prophase. So my chromosomes are going to decondense, they're going to go back to their spaghetti chromatin form. I'm going to get rid of my spindle, it's done its job, it's already pulled the chromosomes apart, I don't need it anymore. And I'm also going to, my nuclear envelope's going to reform. I'm going to start to make two distinct nuclei. And at this point, the division of the nucleus, or mitosis, is complete. So here you can see a cell going through telophase, and you can tell it's telophase because I'm starting to really form two distinct nuclei. Same thing here as well. So let's summarize what we've done so far. So first we had interphase, where the cell grew, it replicated its chromosomes, it made two copies of the chromosomes. And then in prophase, my chromosomes, they condensed, they went from the spaghetti form to their chromosome more condensed form, and here the blue part is the chromosomes. Also in prophase, my spindle, that green thing, started to form. In metaphase, my blue chromosomes lined up right down the middle of the cell. In Anaphase, they were pulled apart. And then in Telophase, I formed two distinct nuclei. So finally, the last thing we have to do is actually split the cell. Right now we have two nuclei in one cell, which we don't want to do. So the word for this is cytokinesis. So basically here, the cytoplasm and all the organelles get kind of split in two, or we make two cells. So in animal cells, this happens by basically pinching the cell in half. If you picture the cell as like a giant balloon, if you tied a rope around the middle and pulled, it would kind of make two little balls. And that is how cytokinesis works in animal cells. This little portion right here where it pinches to form two cells, we call that the cleavage furrow. So if we look at the picture here, this is an actual EM microscope picture of a cell undergoing cytokinesis, and you can see we're getting that cleavage furrow to pinch right in the middle to create two cells. So if you look at here, this picture of cytokinesis, we've gone from telophase, where we have two nuclei in one cell, and then during cytokinesis, we pinch down the middle to make two daughter cells, each with its own nucleus, an own copy of the DNA. The only thing else I have to tell you is that cytokinesis looks a little different in plants. Because plants have a hard cell wall, we can't just pinch down the middle. It wouldn't work. That cell wall is too hard. So instead of pinching down the middle in a plant, instead what happens, so I'm going to draw, so here's my plant cell that's already undergone telophase. So it has two nuclei with chromosomes in them. So I can't just pinch my plant cell. So instead what happens is new pieces of cell wall form right down the middle. And we call that the cell plate. So now I have two plant cells, each with one nucleus. So this looks a little different than in animals. In animals we just pinched in the middle. But here I just build some new cell wall down the middle to create two new cells. The other difference in plant cells is plant cells don't have centrioles, that organelle that built the spindle fiber. But instead, they can still build a spindle. They just have to use different enzymes to do it. They don't use that organelle called the centrioles. Okay. So that's it. So here's a picture summarizing the cell cycle or mitosis in plant cells, but we have talked about how the cell divides now.